Boltz-2 is a recently published open source model that combines protein structure prediction with binding affinity estimation. Following its publication, there was substantial hype around the model because of its promise that it could lead to significant advances in drug discovery. In this video, we will answer the question, how well can Boltz-2 predict substrates for enzymes? This is an important task in biotechnology. For example, because if you want to produce a drug molecule or another valuable compound, you often need to find an enzyme that can catalyze a specific small molecule. So the question is, can Boltz-2 help with that? Boltz-2 is an advanced open source AI model designed to predict how biological molecules such as proteins, DNA, RNA, and small molecule ligands interact and fold together in 3D space. It builds on the foundations laid by Google's DeepMind's AlphaFold models. AlphaFold 3 can predict the 3D structure of many different molecular complexes very accurately, but its main focus is on the final folded and bound structure. Boltz-2 is an alternative with similar structural prediction capabilities, but it also goes one step further. In addition to predicting structure, it also predicts how strong different molecules bind to each other. That is exactly why it is interesting for enzyme-substrate prediction. A small molecule first has to bind to an enzyme before it can be catalyzed by that enzyme. So even though Boltz-2 was not designed specifically to predict enzyme substrates, it may still capture part of the signal that matters for this task. To predict binding between a protein and a small molecule, Boltz-2 uses what is called an affinity head. This takes the internal representation of the predicted protein-ligand complex and translates it into two scores. The first score is the affinity probability. This is a value between 0 and 1, and it works a bit like a yes-no filter. It tells us how likely is it that the small molecule is a true binder of the protein. The second score is the affinity value. This is a quantitative prediction of binding strength, usually expressed as PIC50, which is a common measure in drug discovery and can be used to rank molecules by predicted potency. For our analysis of Boltz-2, we focus on the affinity probability predicted by Boltz-2. We also tested the affinity value for enzyme-substrate prediction, but in our experiments it gave worse results. So for the rest of the video, we will use the predicted binding probability. The task in this video is the following. Given an enzyme and a small molecule, predict whether the small molecule is a substrate, yes or no. To evaluate this, we use the ESP test dataset, which contains experimentally confirmed enzyme-substrate pairs. One challenge here is that the literature contains far fewer confirmed negative examples than positive. Negative means enzyme small molecule pairs where we know that the small molecule is not a substrate. Because of that, the dataset used sampled hard negatives. This is a reasonable approach because if we randomly pick a small molecule for a given enzyme, the chance is very high that this is not a true substrate. In this dataset, for each positive enzyme-substrate pair, 3 negative non-substrates were sampled. So we have a positive to negative ratio of 1 to 3. Overall, this gives us around 13,000 test pairs that we run through Boltz-2. If we first look at the overall predicted probabilities for the two classes, we see that for the more than 9,000 negative data points, Boltz-2 predicts an average affinity probability of about 42%. For the roughly 3,000 positive data points, the model predicts an average affinity probability of about 50%. So this separation is not very strong, but it is still encouraging that the positive class gets a higher mean predicted probability than the negative class. So there is at least some useful signal. To put that result into perspective, we compare Boltz-2 with the ProSmith model. ProSmith is a multimodal transformer that processes both the protein and the small molecule together, models their interactions, and predicts whether the small molecule is a substrate for the enzyme. So unlike Boltz-2, this model was trained specifically for enzyme-substrate prediction. It was trained on the training split of the ESP dataset and then evaluated on a completely unseen test set. So this is a task-specific, supervised model, while Boltz-2 is better viewed as a zero-shot approach for this problem. And when we compare the two, we see that the model trained directly for enzyme-substrate prediction performs clearly better than Boltz-2. To quantify this, we use the Matthews correlation coefficient MCC. This is similar in spirit to the Pearson correlation coefficient, but for binary classification. A value of 0 corresponds to random predictions or to a model that always predicts the same class. A value of 1 would correspond to a perfect classifier. For Boltz-2, we get an MCC of 0.18. So this is not a very strong classifier, but it is still better than random. We see that the performance metrics for ProSmith are much higher. At that point, I wanted to look a bit deeper into the scores. Maybe the absolute affinity probabilities are not well aligned across many different enzymes. In other words, maybe it does not make that much sense to compare scores between enzyme A and enzyme B. But maybe the model is still useful within one enzyme, meaning that it can rank candidate molecules for the same enzyme in a meaningful way. So I asked a slightly different question. For each enzyme, does the model assign the highest predicted probability to the true experimentally verified substrate and lower probabilities to the 3 non-substrates? This is a more enzyme-wise ranking view of the problem. Here, Boltz-2 actually looks a bit better. In almost 50% of the cases, the model assigns the highest probability to the true substrates. This is a nice result because with 1 positive and 3 negatives per enzyme, a random predictor would only be correct in about 25% of the cases. So Boltz-2 is clearly better than random when it comes to ranking candidates for the same enzyme. But again, if we compare this to our trained ProSmith model, the difference is still very large. ProSmith assigns the highest probability to a true substrate in more than 94% of the cases. So even in this enzyme-wise ranking setting, the task-specific model performs much better. Next, I wanted to analyze how much the performance depends on substrate similarity to the training data. This is important because both approaches may benefit directly or indirectly from having seen related molecules before. For the trained ProSmith model, this is especially relevant because if similar substrates are present in the training set, the model may generalize more easily to new enzymes paired with those kinds of molecules. To measure this, I computed molecular similarity using Morgan fingerprints. These are binary fingerprint vectors that encode structural features of small molecules. Then I used the Tanimoto similarity to compare each test substrate to substrates from the training set. Based on that, I divided the test molecules into 4 groups. The first group contained substrates that were already present in a training set, just paired with a different enzyme. The second group contained substrates with a similarity between 66 and less than 100%. The third group contained substrates with a similarity between 33 and 66%. And the fourth group contains substrates that are very dissimilar to anything in the training set with similarities below 33%. What we see is that ProSmith performs much better when the test substrate is similar to substrates from the training data. That is not surprising. If the model has seen similar molecules during training, it becomes easier to generalize to new enzyme-substrate pairs involving related compounds. For Boltz-2, in contrast, this effect is much smaller. And that makes also sense, because Boltz-2 was not trained on this enzyme-substrate prediction task, so it should not benefit nearly as much from similarity to the training substrates of our benchmark. There is still a slight increase in performance for more common or more familiar substrates. A likely explanation is that Boltz-2 works somewhat better for small molecules that are well studied and broadly represented in biochemical data. But overall, the effect is much weaker than for the trained model. And what is also important is that even for substrates that are not very similar to the training molecules, ProSmith is still slightly better than Boltz-2. Once we move to substrates that are more similar to the training data, that advantage becomes larger. So overall, this benchmark suggests that Boltz-2 is not really the best choice for predicting enzyme substrates. Compared with deep learning models trained specifically for enzyme substrate prediction, Boltz-2 is not competitive with the current state of the art for this task. However, Boltz-2 was not designed specifically to identify new enzyme substrates. Its main purpose is to predict molecular structure and protein-ligand binding. So it makes sense that we still see some correlation with enzyme-substrate relationships, because substrates first need to bind to the enzyme. This means Boltz-2 captures part of the problem, but for accurate enzyme-substrate prediction, task-specific models still perform much better. At Protein Vision, we develop enzyme-substrate prediction models on much larger datasets than those used in currently open-source state-of-the-art models, and we also work with more advanced architectures. So if you are interested in using a highly accurate model for predicting enzyme-substrate pairs, or if you want to fine-tune such a model on your own data and would like support with that, please get in touch with us at info@protein.vision.